Thanks, Michelle. So welcome everyone to the Middle Rio Grande Endangered Species Collaborative Programs Seminar Series. My name is Catherine Murphy and I'm the Science Coordinator for the Collaborative Program. Today's speaker, Dr. Charles Yackulic, will present recent work on the development of an integrated model that informs flow and non-flow management impacts on the Rio Grande. He and his co-author began this work with the Population Monitoring Work Group under contract with the New Mexico Interstate Stream Commission, and have continued graciously the expert elicitation exercise with the collaborative program as a science and technical statistician in Flagstaff, Arizona, sharing this important work with the collaborative. If you'd like to unmute yourself at that time after Charles' presentation to ask your question, you can, or feel free during the slides to type in your question in the chat, and we'll read those out at the end. So without further ado, I'll hand it off to Charles.
Great. Thanks for that introduction, and thanks everyone for tuning in. I want to start, let's see. I just want to start with thanking a lot of different folks. You know, most of who I tried to write down here, although there's a lot of other folks who have contributed to this work over time. My co-authors on the manuscript that's about to be submitted on this work have all been super helpful in helping me understand the system, understand the data. We've been working on this for a bunch of years as a collaborative sort of project with lots of sort of an iterative approach in which, you know, took a first stab at it, and then folks would either point out other data that they'd like to see integrated or concerns they had or issues, and went through a lot of iterations to get to where we are, which I think is, you know, it took a while to get there, but I think it's been worth the effort to get there. I also really want to thank the New Mexico Interstate Stream Commission and the Albuquerque Bernalillo County Water Authority who funded this, and all the agencies and the folks that contributed to this collaborative work. So anyway, the outline for my talk today is I'm going to begin by discussing some of the motivation for developing this model. Then I'm going to talk through the model structure and some of the data that were used in it. Next, I'll move on and talk about some of the results. And lastly, I'll get to some of the management implications and sort of how we assess the effectiveness of various management actions. So why build a model? This is actually a slide that I presented to the population monitoring group 2 or 3 years ago when we were first starting this project. And so, you know, I think some of the reasons I think of building a model are primarily because we want to predict outcomes of management and the associated uncertainty. You know, we want to help managers make better decisions and understand, you know, the implications. But models are also useful for evaluating multiple competing hypotheses. There's often different ideas about how a system might work, and they're a way to formalize those different hypotheses and evaluate them. And lastly, they can be useful in determining monitoring and research to reduce uncertainty or discriminate amongst these competing hypotheses. So why build a model collaboratively? I think a group can develop and operationalize multiple competing hypotheses better, but maybe not quicker than one person. And so I work a lot in the Grand Canyon, and this is something that I've come to realize over time through my work there, is just working on these projects collaboratively leads to better products. It's often not as quick as if I just sat down and did something by myself, but I think the product is better in the end from working with a bunch of folks, doing a lot of talking and listening to understand, you know, for the people that really understand the system better, what are their mental models and how can we sort of change that into a statistical or mathematical model? I think it also increases transparency and it minimizes unintended assumptions. I think when you go into a cave and you model by yourself, you can make a lot of assumptions without even really thinking about it. And that's why, you know, having a group that's sort of always checking you and pointing out different ideas is really useful, I think, for the final product. And then why build an integrated model? Well, in this system, there are lots of different types of data being collected, either through monitoring or through various research efforts. There's, you know, ACER CP catch per unit effort monitoring. There's the rescue data, which is actually a form of actually a pretty useful form of data. There's the riverized data, which is looking at drying. There's been various mesohabitat availability surveys, which are useful for interpreting how much pool versus run riffle habitat is available. There's been a lot of work on flow relationships between flow and larval habitat. There's been reproductive work in the field, and also there's been work in the laboratory. And in addition, there's lots of ongoing management. It's not like we're just thinking about one or two management actions. There's various different management actions that folks are doing. So trying to synthesize these all into, you can look at one piece, or you can try and synthesize this into one model that kind of allows you to look at the various pieces together, hopefully. So, that's sort of the broad motivation for why we started this work. And now I'm going to go a little bit into the structure of the model that we developed. And I will say, as I mentioned, this is an iterative process. So this is a little bit different than where we started, but I think it kind of makes sense for the data and the questions we were asking. So some of the basics of this model. In the model, there's 3 river segments: Angostura, Isleta, and Sanakasha. And there's 4 age classes. There's age 0 fish, there's age 1 fish, there's age 2+, and there's augmented fish. And some of the key processes in the model are assumed to occur on April 1st, July 1st, and November 1st. And I'll get into some of those processes what those are. But for example, on April 1st, that's when we determine how many effective spawners there are to produce eggs and to create, age 0 fish. And then we model the larval habitat availability, but then we don't actually start comparing the abundance of age 0 fish to the catchment unit effort until July 1st. So that's sort of when, from the model's perspective, age 0 fish show up is on July 1st. And then on November 1st, we kind of switch from a over summer survival rate to an over winter survival rate. And it's also typically when soon after there's when augmented fish are added. We make daily predictions though between these key dates. And those predictions are primarily based on natural mortality rates, which are estimated, but also include things like the influences of drying and rescue. And these predictions are compared to monitoring and rescue data, as well as other forms of data. And we use the daily discharge at either Sanakasha for the Isleta and Sanakasha segments, or Angostura for the uppermost segment, to adjust parts of the observation process. And I'll get into sort of how we think discharge affects various aspects of the observation process a little later. And another key thing is that the 4 population abundance estimates that ACER did between 2009 and 2012 are really important for this modeling work, for allowing us to make sort of estimates of absolute instead of relative abundance. So this is one way to, think about the population processes as they're represented in the model. So these gray bars, their height is supposed to represent in a very conceptual way the abundance of that age class. And then on the x-axis, we have time in terms of the different seasons. So as I mentioned, we're determining the number of spawners in April 1st, and that's the arrows pointing from the various different triangles down to the spawner N are trying to illustrate that. And one key thing is that we don't assume that all fish contribute equally to the effective spawner. We put priors on this based on some laboratory studies and based on the average size of fish. And essentially we assume that age 2+ fish produce about twice as many eggs as age 1 or augmented fish. Although there is opportunity for the model to push back on that assumption because it's not a fixed value, it's what a Bayesian would call a prior. We also have this larval carrying capacity, which I'm going to spend a lot of time talking about. And that's related to flow, but it's integrating some expert elicitation we did to try and figure out how should we translate the daily flow estimates into sort of an idea of how good a year is. And I'll talk a lot more about that. The combination of that spawner abundance and the larval carrying capacity are what determine the amount of recruitment there. So you can see the recruitment, it starts out as a pretty wide gray bar. I guess I'm assuming it's a good year here. And the difference between that dark gray bar and the lighter gray bar is sort of the influence of drying that's assumed to be occurring, which obviously varies between years, and we account for that. Other things that are in the model are we account for the sort of the augmentation. That's supposed to be what the purple arrow there is indicating. And so again, just diving into what's happening in that springtime is we're taking the daily time series of flow at different gauges, and we're looking at relationships between larval habitat and flow, but also thinking about things like the duration that larval habitat is required, the timing, and other aspects of the larval habitat. And we're using that to create this larval carrying capacity index, and that's linked to the spawner abundance to predict recruitment. And so just a little bit more about that. We're using what's called a Beverton-Holt relationship. This is a relationship that's been around in the fisheries literature for, going on 60, 70 years now. You can imagine it as sort of a steep curve at lower numbers of spawners that then plateaus. So in this example to the right, that's actually an example that was fit for a couple of years. And if you were to keep going over on the x-axis, those curves would flatten out. And so you get to a point at which the number of eggs produced, i.e. the number of spawners, is not what's limiting your age zero production. And the difference between that green and that blue there might be the amount of larval habitat. So these are 2 years that actually had pretty good larval habitat. The green one is 2019 and the blue one is 2017. And this is just showing how, even though you had more larval habitat in 2019, you had less effective spawners. Now, all these things, I'm showing them here as dots, but there's a lot of uncertainty in all these relationships, and that's pretty important. So in order to, estimate this larval habitat availability, we wanted to answer some questions about, or we had some questions that would help us construct that, including, how is larval habitat availability related to discharge? This is a question a lot of people thought about in the program. Another one is, how long is habitat needed for larval development? When is it needed? And what are the cues for spawning? And we took an approach to addressing those of what's called expert elicitation. So 5 of the folks who are co-authors on this went through this exercise of a long Excel spreadsheet where we were trying to get them to estimate things like, well, this one, we've got a description at the top, and for each of these, we had a pretty long description that we all talked about to make sure that we were talking about the same thing. And then in this example, they're trying to elicit, when do they think, in a year when they're not limited by flow, when do they think that silvery minnow would produce eggs? What would be the timing of spawning if there was no limitation of sort of its of water? And so, each of the experts went through this exercise, and we use what's called the Delphi process. And the way that works is you have folks, for anything that you're asking them to think about, you have them start with estimate a lower version. So that's a lower confidence interval, lower CI, that column to the second column. Then you have them do the upper. Then you have them try and estimate the probability that truth is somewhere in the middle of that. And then at the end, they give you their best guess. And so everybody did this for a number of different metrics. And then we met and we discussed, and people went over, different sort of ideas, and people discussed why they put something versus another person. And then the 5 folks went back to the drawing board and they assessed whether, they wanted to change any of their values based on the group conversation. But the key is sort of that the experts did their filling out of this alone so that they weren't influenced too much by strong personalities in the room. But there was time for discussion and learning from each other. Another key part of this model is what happens in terms of drying. So drying is affecting all the life stages that you can see with the light gray bar here. And I'm not going to go through the math of this so much as looking at how did we figure out how much drying was affecting the abundance on a given day. Well, in this case, this is for age zeros, that equation on the bottom there. And so you've got your production on July 1st. That second component there is looking at the natural mortality, that second part of that first thing. And so that's sort of how much mortality should there be occurring just as background for this species. Then we have the proportion of the reach that is not dried. We assume that if you're in the part of the reach that hasn't dried, you've been unaffected. So the only thing that matters for you is this natural mortality. We then have the proportion of fish that were exposed to drying after July 1st. And we assume that age zero fish cannot escape drying before July 1st. But after that, they could. And this tau parameter determines how many fish move out of the reach as it's drying. And we estimated that through expert elicitation, although I also looked at afterwards at the extent to which different values might change results. And they didn't have huge impact within sort of the range people thought this could be. And then this last part here deals with, if you don't move, that's the 1 minus tau there. What's the probability you're rescued and you survived? And we used some of the recent research from Thomas Archdeacon and others to parameterize this based on sort of the timing of when rescue occurred. And so for age zero fish, we assume before July 1st, if you were in a dry area, you did not survive it. But for age ones, that assumption did not occur. And this is just something, this is based on a paper that Thomas and others put out in 2020. We took some of the data and there's fit a relationship about how rescued fish survive based on the number of days after April 1st. And then lastly, the augmented fish, we assume that they're subject to a stocking survival, that there are, and this is a pretty standard assumption whenever you put fish that have been in a hatchery into the wild, typically a large number of them die, within the first couple of days or weeks of stocking, and then, some proportion survive, and then we assumed after that they behave like wild fish once they got through that initial bottleneck. So in order to link this model, which is modeling abundance, to the actual catch data, which is the majority of the data that we had, we kind of used a version of this equation here. Now, I try not to put too many equations in my talk, but I'm going to walk through this one just because I think it's important to understand. So we're estimating N abundance of a particular age class. So it could be age 0, age 1, in that river segment. So you could imagine that that's Sanakasha age 0 fish. Well, then we're going to multiply that by the proportion of the river segment population that's in a site. So when there's drying that's occurred, more of the population is going to occur in each wet site because they're scrunched into a smaller area. And so we figured this out based on the number of wet river length on a particular day. And then the size of the site in terms of monitoring, which would always be 200 meters, or for rescue data, how long an area they rescued in a given day. And then this F is the proportion of fish in the site that were sampled. And so this is sort of looking at, okay, if you're talking about ACER monitoring, there's the 200-meter site. We've multiplied by this first theta to get to sort of, okay, how many of the fish should be in that site? But now we know that you don't sample the whole site. You only sample a portion of the site. And more to the point, the sampling often is preferential to pool mesohabitat over run riffle. And depending on the flow, we know that there tend to be more fish in the pools than in the run riffle habitat. So we're trying to account for that. So we're focusing on the proportion of fish in the site that were likely sampled based on how much sampling occurred in the two mesohabitats and how much was available. And then lastly, even if you get down to areas that are seined, we know that if you've ever spent time out there trying to catch seine for fish, you don't catch every fish that's there. And so the P is really dealing with the fact that you miss some of the fish that are there, even if you seine through an area where there are fish. And just to this point of how discharge affects various processes relevant to translating abundance to catch. On the left here, the blue is the percentage of pool that we estimate are in the river at different discharges. So notice that the x-axis is logged here. So you're going from a discharge of 1 CFS to 10 to 100 and then to 1,000. And then on the y-axis, you use the right axis in that left pool. In this one right here. And so pools are about, roughly 5% of the river segment at lower discharges, but it declines quite a bit lower as discharge goes up. And this is important because typically ACER samples between 10 and 25% pool. So they're sort of preferentially sampling that type of habitat. So we wanted to account for that in the way that we looked at the data. In addition, this one on the right here just shows how the preference of fish for pools relative to runs riffles increases with discharge. And this is something you can sort of get at just by looking at some of the raw data. If you pair samples, there's a lot of fuzz when you just look at individual samples, but this is the relationship when you look across all the samples. So basically, if you're below about 50 CFS, there's actually, there's not much preference, or there's a slight preference for the runs and the riffles relative to the pools. But if you're over 50, there's an increasing preference for the pools. And you can think of this as sort of as the runs and riffles probably get more and more velocity, silvery minnow are not spending as much time in them. Okay, well, I sort of cheated there. That was a little bit of a beginning model result, but I think it was important for understanding how we link the observations. And now I'm going to go more fully into some of the results of this process. So I'm going to begin by talking about the expert elicitation. One of the keys here is that we did this without looking at any data. None of these were fit to data. This is just us going through that exercise of what different people think. We later used that to say how well does it predict what's in the data, but these relationships were asking specific questions based on expert knowledge. So in this particular one, this is looking at the timing of when we think eggs are laid over the course of the reproductive season. And generally speaking, different experts agreed. So here, each one of the different colored lines is the mean expectation of each expert. And then the black line there is actually the best performing one when we went to actually compare them to data. And what we did was for each expert, we considered both their mean, plus we considered changing it so that it was either a little bit later or a little bit earlier by emphasizing the parts before or after May 1st. So if you compare the black to the blue, that's the expert who ended up having the best performing one. And their best performing was to shift a little bit more of the mass for reproduction to after May 1st. And that was consistent across all experts, where actually shifting these curves to put more emphasis on reproduction later in the season generally led to better fits to the data than people's mean expectation. Folks tended to agree on the duration of inundation required. And so these are the different experts again. And interestingly, all of the experts except for the green one there, their model performed better when you went to the lower standard error there. So that's where that black dot is for that particular expert. But if I were to plot this for each expert's model performed better when it was lower. And they sort of converge on this common idea of 20 days of inundation being around what's required. There was some disagreement about Qs, but really there wasn't a lot of information in the data too. So versions where you took each one of the experts and you either took the lower, the mean, or the upper did not have a huge difference in their ability to predict recruitment. And so this one just seems to have a lot less data to inform it. And lastly, there was disagreement among experts regarding how much larval habitat was available at different discharges. And so we actually did this for each of the 3 reaches, but most experts didn't have huge differences between reaches so much as they just had fundamental differences about what they thought this relationship was. In this case, you could argue that in a qualitative sense, 4 of these experts have pretty similar expectations, whereas one of them had this idea that there was potentially a lot of larval habitat available at low discharges. And again, the black line is the relationship that performed best when we calculated this larval carrying capacity index and compared it to the data. So we put together these various relationships into this index, and then we looked at how well did it explain variance in the larval carrying capacity. And so each one of the dots here is for the means of each expert, and then the black is for that best performing overall. And for comparison's sakes, I also fit in the model where instead of going through this whole expert process, we just took the May-June mean flow, which is an index that's been shown to perform pretty well in the past and just sort of in correlative regression models. And so you can see that actually that model does pretty well in Sanakasha. The best model from the expert elicitation does a little bit better, but they're pretty darn close. But where it really shines is it's sort of in Angostura. Our end goal for this exercise is to do better prediction. And so we fit this model using data from 2002 to 2018. And then in order to see how well it was doing, we tried predicting 2019 and 2020 October catch. And one key here is I think that this exercise would be useful, as long as it does roughly as well as some of the other options out there, just because it might help us think about the processes. But if it did a lot worse, then we'd have to scratch our head and go back to the drawing board. And in this case, it seemed to perform, as well or better than the 2 alternatives we looked at. So in the left column there, that's the prediction, where the darker is the 10th to 90th quantile, the 80th percent CIs, and then the light ones are the 95%, and the dot is the mean prediction. And then the red dotted line is what the catch that was observed in 2019. In the middle, we have this integrated model, which is the structure is the same. We're just using mean made through June discharge instead of this expert informed covariate. And then on the right, we have the regression model and this is the regression model from the biological opinion, which is just based on flow. And so, in 2019, the integrated model with the expert informed covariate does better than the other 2 options. I think part of that gets back to this. I think 2019 was a somewhat unique year in the sense that there was spawner limitation likely in that year. And I'll talk about that a bit more. In 2020, the models, perform, I think, a lot closer to better. And that makes some sense to me that these other models might do nearly as well or almost as well. But my take home from this is the model is doing better in 2019 than the 2 alternatives and probably about similar in 2020 in predicting data that none of these models had ever seen. We also did a series of checks to make sure the model was repeating some properties of the actual data. So in the top rows here, these are just observed versus predicted on a log+1 scale, just so you can see all the data. And they tend to agree pretty well. Each one of those dots is a different ACER sample or a different day of rescue data. So there's quite a lot of data coming into this. And then on the bottom there too, this was kind of a key one. Our model does not include any zero inflation, which is different from I think just about all the past work that's been done in the system where people separate out the zeros. And so we wanted to make sure that this model would reproduce the same amount of zeros as was in the data. Because if it wasn't, that would be evidence to us that we have to kind of do what other folks have done and have this zero inflation. But so the gray bars here are sort of simulations from our model based on how many zeros should it think that there will be. And then the red dotted line is how many are observed. So in both cases, we're not seeing any more observed zeros than what the model predicts, and it sort of falls within that distribution. So that makes me think that we don't need to add the additional complexity. And so, what that means is we're using a negative binomial, and a negative binomial predicts zeros occasionally when you have really low abundances. And so that's the explanation, is that we can get to those zeros just by saying, hey, sometimes you shouldn't expect to catch a fish, even though there's some fish there, because there might only be one per 100,000 meters squared, and you only seeing 10,000 meters squared. And so, getting into some of the more biological results from the model, not surprisingly, the model suggests that there's substantial variation in minnow abundance over time. So here in the x-axis, we have different years, and in each year, the green is the abundance of all age classes of wild-born fish in the spring before reproduction. And then the orange is the fall abundance of all the wild-born fish. And then for contrast, I have the augmented fish. And so you can see what folks who are in the field, already know, which was in years like 13, 14, and 15, more fish were augmented than not when they were out doing sampling in the spring. We can also see the extent to which the model is agreed with the population estimates. So those in 2008, 9, 10, and 11 there, you can see the estimates in terms of the dot and the 95% confidence intervals, and they line up pretty well. Now that was data that was used in fitting the model, so that's not a great check on the model, just so much as to show that it's sort of fitting the data it was calibrated to get from relative abundance to absolute abundance. We also see substantial variation in the population structure over time. So here, each one of these bars is a proportion, and you can look at the colors to get an idea of what proportion of the fish are age 1 or age 2 plus in the spring or augmented. And so you can see, like in a year like 2002, there's a lot of older fish predicted to be in the population. You get into 2003 and there's a lot of augmented fish. And similarly, you can see similar things in 13, 14, and 15. Likewise, in the fall, you can look at age 0 versus age 1 plus fish. And the model also suggests some interesting pattern in terms of survival. So here on the y-axis, we have survival, and it is a log scale. So some caution, some of the differences actually might be bigger than they look in this one. And you can see estimates with and without drying for Sonocassia and Isleta. We also have a Angostura, but obviously they're the same because drying was observed during this period. And these are the mean estimates over the whole 2002 to 2018 time period. We also have a figure in the appendix that shows year by year ones, but there's a lot of uncertainty in each year's estimate. Some of the interesting things to this, you look at Sonocassia and the model seems to estimate, quite a bit higher age 0 survival and overwinter survival, but quite a bit lower age 1 plus survival. And this is interesting, and it does make me think about some of the work that Mike Hatch and others have been doing, thinking about age truncation, whether that is, particularly an issue in Sonocassia. Isleta and Angostura have, much more similar vital rates. And then generally the larval carrying capacity index tracks this mean May and June discharge, but there are some important differences. So here on the x-axis, for each year, I've just calculated the mean May and June discharge, and then on the y-axis, we have this larval carrying capacity index. And so, they're both of these metrics do a fairly good job of predicting, as we saw in the R squared. But the larval carrying capacity index really does a lot better in Isleta Angostura, and a little bit better in Sonocascia. And so the reason they're performing differently is partially because of, the fuzz in this relationship right here. Years like over on the left, you can see how there's some years where it's up near 0.5 and other years where it's down near 0.25 for similar amounts of water. And those get into some of the subtleties of the hydrographs under those conditions. And as I mentioned before, I sort of teased this age zero production. It generally tracks larval carrying capacity, except in a few years where there's spawner abundance limits production. And so on the left here, we just have hydrographs from two pretty similar years from the perspective of the hydrographs, 2017 in blue and 2019 in green. And then on the right, we have sort of where the model thinks you were on those spawner recruit curves. And so, the model thinks that the green hydrograph is actually better. That's why the plateau is higher for that one. But it was more limited by spawners. And the other thing to note about this is most of these figures, when you make them, are fairly flat. You're out on the right-hand side. And this is the year steepest of the ones that I've looked at. And that has some implications too for why the predictions about a sample were so uncertain. It's partially because you can imagine here that any uncertainty in X, the number of spawners, because we never know that exactly, is going to translate to a lot of uncertainty in AGO production, just because this relationship is so steep. In contrast, for a lot of other years, for example, even 2020, you're further out onto this curve to the right, and the uncertainty in the effect spawners isn't really having nearly as big effect, and you're more just, your predictions are the uncertainty is based on the uncertainty and how much larval carrying capacity there is. Okay, so I've talked a lot. And I'm mostly focused on the biology, but now I want to get a little bit more into the management, which is I know what a lot of folks are interested about. And so part of the motivation for this whole exercise and for developing the carrying capacity index was to be able to make more specific predictions about how changing a hydrograph should change age zero production. If you're kind of basing things on May to June, then that suggests adding water anywhere in May to June should help. Whereas, we wanted to be able to target things a little bit more to provide, some more potential help to managers in thinking about how to add water when it is available. So in this case, you look at, we have two different approaches. So in the lighter green and the lighter blue, that is adding water between May 16th and June 9th. There's a typo there in the figure. And that's based on scenario 3 in the Walsworth and Booty paper. That was one of their scenarios that they looked at. And so in that case, what you do is you just evenly add water to the hydrograph between those dates. And so that's adding the same amount of water between May 1st and June 31st, as a lot of other ways you could add it, right? It's just adding 10,000 acre feet. In contrast, we developed this algorithm which tries to optimize the impact on the larval carrying capacity index. And so it's sort of saying, given this hydrograph, where should you add 10,000 acre feet to max out on the larval carrying capacity index? And what it comes up with is for 10, it comes up with that darker blue. And so what it's doing is it's really filling in the base flows in between these peaks to maintain larval habitat during those periods. And the green, that's what it would do if it had 30,000 acre feet. And on the left there, you can see what it terms it thinks is happening in terms of this carrying capacity index. When you add it just between May 16th and June 9th, you have pretty marginal increases in this index. On the other hand, when you specifically add the water to maximize this index, you can get quite a bit bigger. You can get up to 0.91 or 0.95 from 0.67. And, we did this then across all years. So that was just 1 year. Whereas in this plot now, we're showing every year. And on the x-axis on the left is this carrying capacity index without adding water. And then on the y is what it is when you either add it near optimally, and that's with the dark blue, or if you just add it between May 16th and June 9th, and that's the light blue. Similar on the right, it's the same plot, but just for 30,000 acre feet. And so, if this larval carrying capacity index is really serving as a pretty good proxy for larval habitat availability, this suggests, there's quite a lot of potential to improve habitat, particularly in drier years. and that, adding water near optimally outperforms in terms of maximizing this index, adding water uniformly, up based on a set number of days. And in most years, these increases in carrying capacity are predicted to translate to greater age zero production. So this is a similar figure here, but now instead of having the carrying capacity index, we have the predicted abundance. And so on the x-axis, that's what, the estimate from the model, what each year looked like. And then on the y, that's what comes out after we make this change to larval carrying capacity index. And, so what we see is there's the most potential to increase relative age zero abundance, because that y-axis is a log scale. So it shows you there's more potential to increase the relative amount of age zero production in these drier years on the left-hand side. But actually, if you look at where you're going from, there's a couple years in there in the middle where you're potentially going from 100,000 to 1 million age zero produced. So there's actually the greatest potential for absolute increases in age zero production in those mid years, those years that are around sort of 100,000 as the baseline. Okay, so that was just thinking about the water part, but we also wanted to assess various non-flow actions. And in order to do that, we had to make a series of key assumptions. So in order to estimate the impact of augmentation on the population, we predicted the difference in fall abundance, adding together both age zero and age one fish, based on whether augmentation did not occur at the levels that it did occur, or that it did occur at the levels that it occurred at. So this isn't saying, if there was a year where they hardly augmented, it's going to be no surprise that there was not much effectiveness of augmentation. This is a retrospective analysis where we are basing things whenever possible on the action that was taken in a given year. With restoration, we didn't have great data, so we used expert elicitation, and we just assumed that 10% of the shoreline was restored, and that's how we made that comparison. For flow supplementation, we predicted what would have happened if you'd add 10,000 acre feet using that near optimal approach. For rescue, we predicted the difference in fall abundance if rescue had not occurred relative to the amount of effort that did occur in that year. And then lastly, for reduced drying, we assume that drying in each river segment was reduced by up to 10%. When I say up to 10%, if the maximum amount of drying that occurred in a river segment was less than 10%, we just got rid of all the drying. So this obviously has no effect on Angostura, but in years when it's wetter and there's maybe only 3 or 4% drying of a whole river segment, we just got rid of drying. In other years, if there was 22% of the river segment dried, we would decrease it to 12% and see how much that impacted things. So yet knowing these definitions is important for then understanding some of the results that we get. And so what do we get? When we look at each year, what we have plotted here on the x-axis are the different years, and then on the y-axis we have the percent of the baseline in terms of this fall abundance. So if it's 100, that means it had no effect. You would have had the same amount of fish regardless if you'd done the action or not. When it's at 100, 120, that means you have 20% more fish, a 20% higher CPE in October than you would have had elsewise. And you'll notice that in a lot of cases here, there's blue arrows on the top. That's because flow was expected to increase the population by more than 140%. Whereas all these other actions more or less maxed out, kind of around 30 or 40% effectiveness. And so, in most years, the flow is coming out as the best one, but there are exceptions. In 2005, for example, reducing drying would have been the most effective management action that could have been done by itself. In 2015, augmentation was really important because there was just so few fish left at that point, and it was more important than flow. And one thing here is we did not look at synergies between these. And so you can imagine there are some synergies, for example, with augmentation and flow supplementation that might occur. The other thing too is, again, I'd emphasize these are based on current levels. If you augmented more fish, or if you were able to increase the stocking survival of augmented fish, that would move up a lot of these green ones. Similarly, if you were to focus on more drying, that would move up some of the orange triangles. So these are just with the sort of assumptions that I spelled out in the previous slide. Another thing that we found is that effectiveness of different actions varies by river segment. So in a place like Sanakasha, when you're in the drier sets of hydrologies, which lead to lower larval carrying capacity indices, adding water would be the most effective thing. So that's the blue dot in the middle there. Blue is for increasing flows, and the middle dot is the most effective, but drying would be, next effective. When you get over into higher flows, it thinks that restoration might be a useful option because you're starting to get into the habitat that you'd expect to be restored. But you can also see there are particular years where it thinks fish rescue is important. So that's 2018, that upper left one there. And then the green, there are years in which augmentation would have been important, even in Sanakasha. As we go down to Isleta, we see a relatively similar picture, although we see that there's more years when augmentation might be expected to be useful. And if we go to Angostura, we see even more years where augmentation is important. And it seems like restoration would be a really important thing, improving the amount of larval habitat in almost all years. That would be more useful for increasing production in Angostura. And so what are some of the implications of this work? What I have shown here on the right, this is an example from the mallard harvest that occurs, which is one of the oldest running adaptive management programs. And in that case, back in 1995, there was debate about which model structures to consider. And so what they did was each year they made predictions. So the circles there, the not filled in circles, are the observed data. And each year, based on the observation, they make a prediction, use that arrow for the next year. And then they just saw how well did different models perform. And over time, what shows you on the bottom axis there is that is the performance of the different models. And over time, they threw out essentially the red and the green models. Those ones were not fitting the data very well. And they started to rely more on the blue and the black ones to guide management. And this actually led to changes over time in what harvest limits were under different conditions, because their management was contingent on what they thought was going on in the system. And so, that's a different example, but I think the model framework we're talking about here, it is creating a framework for generating testable hypotheses, because that's sort of the key for adaptive management. You need to make predictions and then see if you do bad or if you do all right. Some other things, I think, some other implications for future research. I think additional abundance estimates in a couple more years would be helpful for model precision. There's only those 4, and it'd be really useful to see how well the model does at predicting that. I think intensive mark recapture at a few sites could provide more direct estimates of survival to see if the sort of interesting differences we saw between river segments are in fact true, or maybe they're related to something that we didn't think about in how we specified the observation process. And then I think the other thing too is that the model framework we developed here as part of this group is something that can be updated as new information becomes available. We updated it most recently as some of Thomas's rescue data became available. We incorporated that in. So it's designed to be modifiable. If people had different ideas about the relationship between flow and larval habitat, we could try to test that or put that into the model as well. So just finishing up here, some of my take-home messages are that working collaboratively, we developed a model framework that integrates various sources of information. I think it provides some new insights, and it predicts as well or better than existing approaches. This framework could be updated and improved as new information becomes available, and I just talked about that. Some other take-homes: in a few years, silvery minnow are spawn limited, but in most years they're limited by larval habitat, and I think that's something that a lot of biologists in the system already knew, but still it just provided some support for that idea. And at model management strengths, based on those assumptions I spelled out, adding water near optimally is predicted to be the most effective action in 15 to 17 years. But under certain conditions, other actions may be more effective. And effectiveness varies by river segment. And we did not consider synergy. So that might be something useful to think about in the future. And with that, I'd just like to thank folks again. Lots of thanks to the people who worked on this with me together. We all started at the population monitoring group, as well as a bunch of other folks who have provided insight. I'm sure there's one or two folks that I forgot to put out here too. Lots of thanks to all the people that have worked on with us for the last couple of years. And with that, I'll take any questions.
All right, thank you so much, Charles. That was really great. I, for one, really appreciate the take-home messages. It's very helpful. You all can feel free to type your questions in the chat or just unmute yourself and ask away.
I'll do one. This is Joel. Hey, Charles, thank you for the presentation.
Thanks, Joel.
Hey, you have a graph, you've shown it twice where it has a little plateau thing going on. Where does that cross the axis of one-to-one replacement? What does that look like as a density?
So you're thinking of the graph of age zero production?
Yeah. And you had it for 2017 and 2019.
Right.
And it crossed the x-axis around 30 to 50,000 fish, perhaps. What is
right.
that?
Well, and that was a decision about how I graphed it. It would keep going down. Yeah. So let's see. That's a good question. I don't have an immediate answer for you right now. But the way to get at that would be to, for your effective spawners, it would be a line, right? And that line would be based on the annual survival replacement rate. And so generally what I would, without doing the hard math, what I'd say is, if you imagine a line that's at 10 times the X, that would be fairly close, but I could give you a more exact number. And the reason I'm saying 10 times is because, survival is usually 10% or maybe less. Maybe it's 20 times because your survival is like 5%. But that's something that's a good question. I could look at that. Maybe I'll give you a better answer offline.
I was getting at, it kind of gets towards a minimum viable population of spawners, right? That's what it kind of looked like to me. Whether that's the right interpretation or not, I don't know. But I was just sort of curious what that value could be without thinking about survivorship, but just what does it take to replace the population? Thank you.
Yeah, thanks. Good question, Joel.
Okay, thanks, Joel. We do have a question from Ryan Gronwald with the Corps of Engineers. What was the time frame for optimal flow?
Yeah, typically, it depends a little bit on what existing hydrograph is about where you should add water. But typically the model wants to add water in mid-June, mid-May through sort of early July is typically when it's wanting to add water. But it does depend a little bit on what your base hydrograph is. In a drier hydrograph, it might come up with a slightly different answer than in a different one. But in terms of the timing amongst most of the experts, most of the spawning is sort of happening later May into June and perhaps into early July. I see a question there. How sensitive is the model to the 10% bank line restoration? That's looking at. So that was an assumption we only worked in when we were thinking about the actual management actions. So let me just share my screen again. And so, depending on the year, the predictions can be quite sensitive to that. So you can see right here, years like a 7, 8, 9, 10, thinks you could have had a pretty good increase. And I'm cutting off the confidence intervals here. That is a fairly uncertain management action, though, because, the way this worked is I assume 10% of a river segment had been restored. And then we use the expected increase in larval habitat suitability that had been elicited from the best expert. And so the expert with the best performing model. And so there's a fair bit of uncertainty there, but it does seem like particularly in mid to more wet years, it's quite important. And, as I showed, that seems to be particularly the case as you move upstream into a place like Angostura, where you're often sort of getting sufficient flows. It's just that, the channel isn't well connected. So I think, for Angostura, it could be a really significant factor for increasing the amount of age zero production downstream. It matters in fewer years. I don't know if that's answering your question, Ari.
All right, Charles, we have another question from Megan Osborne. How do you think the results of your management actions analysis would change if you removed flow as one of the measures?
Well, yeah, thanks, Megan, for the question. Based on the way we've set this up, there weren't these synergies, you can kind of just actually just get rid of the blue in this figure, and that would give you an idea of, of what would be most effective in each years. And, a lot of years restoration looks fairly good, although again, it's highly uncertain. Drying looks like it's really effective in quite a few years, that reducing drying would be a great way to increase the population. But you do see there are occasional years where other actions pop up. Augmentation is really important in some years, particularly 14 and 15. And I think if we'd done 19 for this figure, that would have also shown up as well. You also see, there are a couple of years on fish rescue, like 2018, that was really important. I mean, fish rescue, part of the reason it doesn't show up in more years is because of some of the work Thomas had done that had showed that you didn't have very high survival of those rescued fish unless you did it early in the season. And the rub there is that, according to the model, if you do rescue early in the season, you're probably not rescuing the age zeros. So it works in a year like 18 when you've had very poor age zero production and you had a good year the year before, because you're saving some of those age one fish through fish rescue. But in a lot of other years, the majority of if you have any decent production, the majority of your population is probably age zero fish, not age one. And if you wait till when they're big enough to rescue, it's kind of, according to Thomas's work, it's too late because the water quality is getting lower and they're just not likely to survive.
Okay, great. Grace Haggerty
Hey,
posted.
Catherine, my question was exactly what was just asked, actually. So it's just that, gee, you can see that flow is with this river system right now is hard to, it just comes up so high that, maybe we do need to look at all those other parameters. But I think that's really interesting that came out in the model. So.
Definitely. Thank you, Grace. Tom Turner asks, how well does Beaverton Holt capture spawner recruit relationships, and is there any way to validate it?
Yeah, it's funny. In a lot of the other populations I work in, where you hardly ever spend any time spawner limited, I'd say it does pretty awfully. Now that I'm, doing it for this situation, I think it does matter for this particular species just because there is such wild fluctuations in abundance. But, we've done it in the past in the Grand Canyon for both rainbow trout and humpback chub. And we don't have, in the case of rainbow trout, they're hardly ever limited by spawner abundance. In the case of chub, there's not a lot of variation in our adult abundances during a record. And so they do pretty poorly. In this system, they seem to do pretty well. And so, how well does it capture the spawner recruit relationship? I think one way of sort of thinking about how well it does is, from my perspective, is that 2019 year. The fact that that's why the model was actually predicting, so much uncertainty, but also the potential for a lot lower numbers than what came out of a flow only model. Because I would argue, one of the ways to say, should you add in the complexity of a Beaverton Holt relationship is, do you need it? Do you need to know what the spawner abundances are to predict the number of recruits, or is it just environmental conditions? Because that's what I see in some other systems, is the spawner abundance part doesn't really matter. But then you look at something like, let's see if I can find it. You look at this behavior in a year like 2019, and even though this regression model did really well, if you look in the biological opinion, it fits the data fairly well. In the vast majority of years, that's a year where it vastly over predicts, and that's because it doesn't know about spawner abundance. So I guess that's an indirect way of saying that, accounting for spawner abundance does seem to be important in a handful of years for the system. Now, that having been said, there are alternatives besides the Beaverton Holt. I know, Tom, the fisheries literature, so there are a variety of other options available out there of different relationships, some more complex, that one could explore. But I guess it seems to capture some of the importance there of that it's not just about the flows. There is a degree to which the amount of spawners matters. Hopefully that answers your question.
Okay, any other questions for Charles? I have one. Oh, go ahead.
Sorry, you go ahead, Catherine.
Oh, well, I just wanted to ask sort of a generic question, Charles. Did anything about this process, working with these folks and going through this information and combining all these data sets, or the results of the model, did anything surprise you?
Well, I'd say this is when I've gone through similar processes in the Grand Canyon, where I actually had my own strong opinions going into it. I definitely have been surprised. That's part of the reason I became a believer in this process. 90% that would come out of the models, I'd be like, shoot, I told you guys in a meeting 6 months ago before we ever did the modeling, but there was 10% of stuff where you'd be like, wow, I guess, on that one, I hadn't thought about that problem as hard as I should have done. In this case, I came in with less understanding of the system. So I've been learning from people so much through the whole process. I can't identify as much of a clear sort of like something, because the learning has just been sort of gradual and continual the whole time. There isn't a sort of, I don't have a good pre-post comparison, but certainly in the past, I can think of a couple things where I made an assumption based on what I thought, and then we did some hard modeling. I was like, oh, I'm wrong. But so.
Thank you. Okay, go ahead, Grace, if you had something.
First of all, I just really wanted to thank Charles for being willing to come into the Middle Rio Grande, which isn't known to necessarily be super easy to work in with the variable conditions that we have and lots and lots of controversy. But he really got this group together, and I think everybody worked really well, and he communicated well with so many people. And I really wanted to thank him for his several years of efforts on this. So big clap for that. Thank you so much. I know you're working on a publication for this, Charles, and when do you expect that will be out?
Well, I'm hoping to submit it by the end of next week, depending on what sort of comments I get from various co-authors. And we've been through a couple rounds, and I think we're getting down to the relatively minor stuff. So I would hope to submit next week, and then, journals, how long it takes, it's a somewhat hard thing to predict, but I actually already started to put this through the USGS review, and I got back comments suggesting people thought it was in pretty solid shape. So I assume it will, go fairly smoothly, but I predicting journal reviewers is probably harder than silvery minnow sometimes,
And also, just for the audience, I know we've been talking about other things we could do with this model. What kind of things do you think you might recommend that the collaborative program and the others in the basin consider using this model for?
Well, I mean, one thing that I would sort of, particularly on the flow side, I think there's still some work to be done to make it more, what do you do when there's uncertainty in the hydrograph that's coming to predict those near optimal conditions? Because we did sort of a retrospective analysis for that. And hindsight is 2020. I have some thoughts on that after going through this about what that might mean, and then setting up some testable ideas there. I think the model sort of would suggest that finding a sort of a flow that you can maintain for more 20 days or more, what is that? How do you maximize that? And how do you trade off adding more days versus adding higher flow? I think those are some things to think about, that would then be testable in years when there is water. That's definitely something that I'm interested. I also think, we think there's a little bit more that could be done to think about, how might we design things? I have not thought too hard about how different designs for monitoring or for, increasing a little bit of effort here or decreasing there, how that might or might not improve precision. We've kind of talked about that a little bit, but it would be good to sort of if we're going to go down that route, to talk with the folks that are collecting the data, some more of the ACER folks about sort of logistics and how you could actually pull that off and those sorts of things. But those are some of the ideas that come to mind about things that could be done next. Yeah.
Thank you.
Thank you, Grace. Anybody else?
Hey, Charles, this is Joel. USGS usually puts on a standard caveat that anything published doesn't represent the agency, doesn't it? That will be included in the publication?
Yeah, it's somewhere in the acknowledgements. Yeah.
Thank you.
Joel, you'll notice too, I did if you work for the USGS, you get really good at putting preliminary do not cite in the bottoms of your slides.
Just add slide numbers and I really, yeah, you got it. You got it.
Okay, I think that might be all the questions. Charles, I just want you to know that you're not quite off the hook, because I'm sure we'll be we'll be wanting to pick your brain a little bit more, if that's even possible in the future. We're definitely going to take this information and chew on it, and I hope to apply it directly to some of the other things that we're trying to do in the program. And it's the way you've set it up here makes that a lot easier for us. So thank you for that.
Yeah, thanks everyone for your time. And for the folks that have been really involved, thanks for all your help. And particularly Grace, thanks for your patience, because I know we did take a couple of years to do this, but I think it was worth it.
It was a lot of fun.
Yeah.
And we'll try and continue it if I can get the funding rolling next year too. So we'll look forward to working with Catherine more on ideas that we could use the model for.
Absolutely. Good work takes time, Charles. All right. Well, thanks everybody for your time. And Charles, we'll just wait and let you tell us when or if we can post this on the portal.
Yeah, I'll get on that. It'll take a couple days, but it'll be all good.
Okay, no problem. Thanks so much, everybody. Have a great afternoon.
Thanks, everyone.
Bye.
Thanks, Charles.
Thanks.